Okay, I'd like to welcome everyone in attendance to the Center of Vaccine Preventable Diseases webinar. Today we've got an all-star cast of immunologists who are coming to speak about COVID-19 immunity and vaccine development. So without further ado, I think we'll get started because we've got a lot on the agenda. I'd like to start with, so my name is Jeff Kwong. I'm the Interim Director of the CVPD, and I'll be the moderator for today. I'd like to start with an indigenous, with our acknowledgement. We'd like to acknowledge the traditional territories of the Mississauga of the New Credit First Nation, Anishinaabe, Wendat, Huron, and Haudenosaunee indigenous peoples on which the Dalla School of Public Health now stands. The territory was the subject of the Dish with One Spoon Wampum Belt Covenant, an agreement between the Iroquois Confederacy and the Confederacy of the Ojibwa and allied nations to peaceably share and care for the resources around the Great Lakes. We would also like to pay our respects to all our ancestors and to our present elders. So just a few housekeeping items. We will try our best to answer as many questions as possible. There are two ways for you to direct your questions. During the webinar, you may use the Q&A function that you'll see at the bottom of your screen. So you can type in your questions and we'll try our best to get to as many as we can. And after the webinar, you can direct any questions you have to the email address for the center, which is cvpd.dlsph@utoronto.ca. Once again, that's cvpd, Center for Vaccine Preventable Diseases, .dlsph, Dalla School of Public Health, @utoronto.ca. So now I'm going to introduce the speakers that we have today. First, we've got Dr. Jennifer Gomerman. She is the Canada Research Chair in Tissue-Specific Immunity and is Associate Chair of Graduate Studies and a Professor in the Department of Immunology at the University of Toronto. Her research focuses on adaptive immunity, autoimmunity, gut immunity, and multiple sclerosis. She has recently published a preprint study that is under peer review on the antibody response to SARS-CoV-2 in the blood and saliva. Next, we have Dr. Jean-Philippe Julien. Dr. Julien is the Canada Research Chair in Structural Immunity, a senior scientist in the Molecular Medicine program at SickKids, and Associate Professor in the Departments of Biochemistry and Immunology at the University of Toronto. His research focuses on antibody structure and function, structure-based vaccine design, and structure-assisted development of therapeutics. And last but not least, we have Dr. Tania Watts. Dr. Watts is a professor in the Department of Immunology at the University of Toronto and Associate Chair of the Post-Doctoral program. She is also Director of the Faculty of Medicine's Cell Sorting and Analysis Facility. Her research focuses on immunity to viral infections and cancer, focusing on T cells. So the format of this is we'll have the three panelists give some opening remarks, and then we'll have some questions for them afterwards. So we'll start with Tania, then we'll go to Jean-Philippe, and then last we'll have Jennifer. So Tania, do you want to start with your opening remarks?
Yes, thank you, Jeff. Hello, everyone. Thanks for joining us. Today, I'd like to tell you a little bit about how the immune system responds when you're first infected with a respiratory virus, such as the SARS-CoV-2 that causes COVID-19. And I'm going to introduce you to the concept of immune memory, how we can remember prior infections, and how that'll lead into a discussion of how vaccines work in the next section. So when you get exposed to droplets containing a virus, such as SARS-CoV-2, they'll lodge in the mucosal surfaces in your mouth or throat or nose, and that virus will attach and enter and start replicating in the airway epithelial cells. Now, almost immediately, your body, cells of your body, sense foreign material and send out alarm signals. One of these immediate alarms are called interferons that interfere with viral replication in the surrounding cells. So we start with a very rapid battle between the virus replicating and getting into more cells and your innate immune system trying to contain it. Viruses often, including influenza, and we think SARS-CoV-2, also combat these early mechanisms, and the fight between you and that virus establishes how much virus gets in for that initial infection. But while this is happening, your adaptive immune system is kicking in. Some of the molecules that the immune system sends out as an alarm, called chemokines, attract many other lymphocytes to come into the region of the infection, cells, white blood cells, such as T and B cells that I'll talk about. And these T and B cells are going to fight that infection. So B cells are a kind of cell that gives rise to antibody-producing cells. And T cells are another kind of white blood cell that comes in two kinds, so-called helper T cells that are going to help the B cells make strong antibody responses, as well as help the killer T cells that I'll tell you about next. The killer T cells actually will go to the site of infection and recognize when an epithelial cell is actually full of virus and acting as a factory to make more virus, and it will kill that to stop further infection. So T cells and antibodies have a complementary role. Antibodies block the virus, bind to the virus outside of the cell, and T cells get rid of the factories, the cells making virus. Now, the best kind of antibody to completely block infection, we call a neutralizing antibody. That antibody can bind to the end of the spike, for example, on SARS-CoV-2, right where it needs to attach to cells to get in. So if you have a neutralizing antibody, you can completely protect from infection. On the other hand, once the viruses get into cells, you really need T cells to eliminate those virus factors. Now, if you've never seen this virus before, it's a new virus, you're not going to have those T cells and B cells specific, or very many specific for the virus. And so the virus initially gets going before your immune system can ramp up and eliminate it. So maybe at the start of the infection, maybe 1 in a million of your T cells and 1 in a million of your B cells has the right specificity to recognize that virus. And once it sees that virus, they actually start dividing. So you actually get many more copies of the T cells and B cells specific for SARS-CoV-2, for example. So after you've eliminated this infection, and we know that people do, they get sick and the vast majority eventually eliminate the virus and get better, what's left behind is a bigger pool of those T cells and B cells that can recognize the virus if it were to come back. So if you get re-exposed, you can actually get rid of the virus before it even mounts a proper infection. The other thing that happens is B cells give rise to some antibody secreting cells that can be very long-lived. And they can actually keep making antibodies for years and years. And for some diseases, such as the vaccine against smallpox, you actually have antibodies that go on for 70 years. So again, we can be protected from reinfection by pre-existing antibodies after the first infection, as well as by memory T and B cells, because you have more of them, they can react much more quickly to a new virus infection. So this is actually the basis of how vaccines work. What we need to do is trick the immune system into think it's having an infection, so it mounts this adaptive immunity and expands T cells and B cells specific for that virus. And that's done in a way that is to take parts of the virus that are not infectious and give it in such a way as to induce an immune response. So I'm going to pass the talk over to Jean-Philippe Julien next, who's going to tell you how this is being approached in different ways for SARS-CoV-2. So thank you.
Thank you very much, Tania, for this immunology primer in the context of respiratory infections. I think, you know, what I want to introduce in this kind of preliminary remarks are the different technologies. What are the tools that we have for making vaccines? So Tania said it very well, that you want to teach the immune system how to protect, how to mount the right type of immunity that will protect the infection, or if you do get infected, how it can eliminate it so that you don't get disease. And so us molecular scientists have different tools in the toolbox to do so. And so one of the most ancient ways of developing vaccine, of course, is to start with the infectious agent itself. So in this case, you would take the virus that gets isolated from an individual, and then in a culture, you can propagate this virus, and then you can isolate enough of it and then treat it with chemicals to inactivate it. And so that consists of an inactivated viral vaccine. And there are currently 2 of these in phase 3 clinical trials that are being evaluated. And my colleague, Jennifer Gummerman, is going to talk about how vaccines are evaluated after me. So that's one of the conventional ways for vaccine development. And some of the successes of this inactivated viruses come from acute infections. So that means it's a virus that wouldn't establish a long-lasting impact on your body, but could be cleared. Another technology that exists is that you might not want to expose your immune system to the whole virus. You might just want to focus on specific parts of it that are associated with protection or clearance. And so you can introduce at the genomic level a specific part of SARS-CoV-2 in a viral vector to deliver it to immune cells. And there's also 2 non-replicating viral vector vaccines that are currently being evaluated in phase 3. And then finally, you could say, I'm not going to deliver the replicated or non-replicated viral vectors, but really just a small component of the virus, the SARS-CoV-2 virus itself, and deliver it in a different way. So we have seen the emergence of mRNA technologies, and there are also 2 of those that are very well advanced. And so I think, the take-home message is not so much the different names for these technologies here, but to understand that when people are talking about the development of a COVID vaccine, there are many distinct technologies from the molecular level that are being developed. And so one of the strengths of this approach, of course, is that we have many shots on goal. Different types of vaccine induce different types of immunity, and some might be better in specific populations. And so a lot of what we're seeing in early clinical trials, of course, is the evaluation in healthy individuals that are in the adult range. But if we're thinking about evaluating in the elderly or in children or in immunocompromised individuals, it's good to have a wide arsenal of different vaccine technologies that might work better in distinct populations. One of the emerging technologies that is also a new tool in a way is the concept of antibodies and how they're deployed. And so people that have been infected and have mounted antibody responses, you can isolate these antibodies, whether a whole serum or specifically the highly potent monoclonal antibodies, and you can use these as prophylactics as well. So you can give them in a context, for example, where there would be an outbreak in a specific setting, and you could deliver antibody ahead of time to people that might be exposed. And so there are 2 phase 3 clinical trials currently ongoing to evaluate antibodies as a passive transfer, not active immunization, but passive immunization as well. And so scientists have a variety of tools that they're evaluating, and in order to fill some of the needs, of course, that exist in the broad community. It's important to realize that there might not be one COVID vaccine, but multiple types of COVID vaccine, and that's not unprecedented. If you think about the seasonal influenza vaccine, there are actually different types, and that's often very useful. And that can also reduce the demand. If there was only one vaccine that existed and everybody needed to take the same one, you can imagine that from a manufacturing perspective, you would need to produce a lot of a single product, and it would put a very high burden on manufacturing capability. Whereas if you have many different vaccine candidates that are emerging from different technologies, the manufacturing aspects will be different, whether you're cultivating a virus and then inactivating it, or making an mRNA molecule or viral vectors. And so you can leverage many different manufacturing sites. And so from that perspective, to make sure that there's a lot of vaccine doses available, these different technologies are also very important. And then lastly, as my colleague Jennifer Governor is going to talk about, it's important to understand the right doses, the right scheduling for the vaccine implementation. And that is specific to the different technologies that you're developing, and something that requires clinical trials to evaluate. And so I think this is a good point, perhaps to stop, and we can continue this in the Q&A, of course, and I'll pass it over to my colleague, Jennifer Governor.
Thanks, Jean-Philippe, and thanks, Tania. You've set me up nicely, and thank you, Jeff, for introducing us. So my job here is to tell you a little bit about how vaccines are tested. This is a very rigorous process that goes through defined phases. The first phase of vaccine testing is what's called preclinical testing. So this doesn't involve human subjects. It happens in the lab. Often we use animal models such as mice or primates. Both are actually being used currently to test the efficacy of SARS-CoV-2 vaccines. And in the lab, we determine how good those vaccines are at eliciting an immune response. So we'll look at the immune response in the mouse model or in the primate model, and we'll establish a good dosing regime that would work in those animal models that might give us a hint on how to best administer this vaccine when it comes to humans. Once we've got data that convince us that this vaccine is going to work, it's going to generate a good immune response and it's safe in these animals, then we take it to the next level, which is called phase 1. Phase 1 are considered safety trials. So really the main objective of a phase 1 trial is to give the vaccine in a small group of people to test the safety of the vaccine and the dose of the vaccine. So the people who get the vaccine are volunteers. They're carefully screened and they're carefully watched to make sure that the vaccine is safe and that there's no side effects. And when we've convinced ourselves that that's true, then the next step is to go a little bit larger and to give, rather to a handful of people, we give the vaccine to hundreds of people. And that's where we can actually split these groups up into different demographics, for example, the elderly, children, etc., so that we can learn whether the vaccine behaves differently in different populations or not. And from this trial, we get additional safety data because the more people you test your vaccine in, the more information you get. And we also get a sense of what's called efficacy. How well does this vaccine work? And what we do is we test the blood from these subjects and ask, okay, did these subjects mount an immune response, for example? But we're really not going to know how these vaccines work how well they work until we do what's called a phase 3 efficacy trial. And that's where you give it to thousands of people. And here's where you sort of watch and wait. You take a population where, especially one where the virus is quite prevalent, for example, in the United States right now, and you would take thousands of people and randomize them into 2 groups. Half of the group, and there's a trial that's just started now where they've enrolled 30,000 people in the United States, and other trials ongoing, for example, in Brazil. So you would recruit 30,000 people, and then you would divide them in half into 2 groups. Make sure those 2 groups look approximately the same in terms of their average age and other demographic features and sex, for example. And then you would administer the vaccine to half of that group, to 15,000 people, and a placebo to the other 15,000 people. And then you would watch and wait and see whether the vaccine is protecting that large population from developing infections with SARS-CoV-2. So they have to be monitored using the test that we use to determine whether someone has acquired SARS-CoV-2. And we look for whether there's protection conferred by giving the vaccine to those people. And an added bonus is that because we're testing it in so many people, is that we're for sure going to see if there are any safety concerns. They'll likely come out, if not in the phase 2 trial, then in the phase 3 trial when we have that many more people. So you get a lot of data from these types of trials. So if the vaccine does prove to be efficacious and it does protect more than the placebo, the group that got the vaccine, then the vaccine is evaluated for approval. And each country has an approval agency. For example, in Canada, we have Health Canada. In the United States, they have the FDA. And they look at all the data and they decide whether the vaccine gives a real benefit to those who received it, and whether it's sufficiently safe to roll out into the population. And if it is, then that's what happens next, is the vaccine is given to large populations and manufactured and distributed. So those are the phases of vaccine development, but some of you might be wondering, why would I even take a vaccine? I've been hearing that I don't even make an immune response to SARS-CoV-2. And I'm here to tell you that there's good news. Most of the data that's coming out of recently, out of labs, and I'm talking last few weeks, have really shown that we can make a good immune response to this virus. Most people make a good immune response. Some people make a dysfunctional immune response and they get sick. So we don't know who's going to be who. And so we want to make sure that, as Dr. Watts said, that when we give this vaccine, we're tricking the immune system. We're telling them, you respond to this in a really safe, controlled way so you can make a protective immune response, so that we don't play Russian roulette and we don't decide, okay, I'm going to just expose myself to this virus, but we know that some people get really sick from this virus and some people die. So we want to make sure that this immune response is done in a controlled way and it's an effective immune response. And so that's why we vaccinate. In addition, when we vaccinate enough people, the hope is that that will confer herd immunity. And so for people who may not respond well to vaccines or who can't take vaccines for various reasons, if we have a sufficient herd immunity through the population, then we'll have protection as a society, not just as individuals. And so that's really important. And we know that to get herd immunity, we're going to need vaccines. We know that in countries that were hit really hard, like France and Spain, when they've done serosurveillance studies, only about 5% of the population have made antibody responses to this virus. And so it's going to take a long time if we rely on nature for us to have herd immunity. And that's why vaccines needed, especially now, to deal with this pandemic. So I think that's all I'm going to say, and I think there's some questions for us. Great.
Thank you so much to Tania, Jean-Philippe, and Jen for your fantastic remarks. So I'm going to start with some pre-prepared questions for each of our speakers, and we will also go on to take your questions from the audience. So please, if the audience, if you have any questions, please type them into the Q&A, and we're going to get to some of these after these initial questions. So first of all, for Tania, can you tell us if there's any pre-existing immunity against COVID-19?
Yes, so there's been several studies that have taken blood samples that were collected prior to the emergence of this pandemic and tested the T-cell, white blood cells in those samples for responses to the new virus SARS-CoV-2. And in about 30 to 50% of healthy people whose samples were taken before the pandemic are able to mount some immune response to small parts of that virus that are conserved between the new virus and some seasonal coronaviruses. The data says it's probably not as strong as an immune response as someone who's just been infected. And we don't know if it's enough to completely prevent infection, but there's some thought that when you see a lot of variability in the population with perhaps 80% only getting very mild disease, it's possible that those T-cell responses are actually controlling the virus quickly And so they're not getting a lot of symptoms, and that may be due to this pre-existing immunity. Of course, that may not be the only reason for differences in individuals' disease severity. There's quite possibly some minor genetic differences between people. So we don't know the whole story, but we think that prior immunity might have some role.
Great. And the next question is, can you tell us a little bit about the difference between the response to natural infection and vaccine-induced immunity, and why might the vaccine give better immunity?
Well, first of all, with the natural infection, you have all the components of the virus. It's a live virus. It's going to start replicating and making many copies of itself. Some of the peculiarities of this particular virus is that it binds to a molecule, that ACE2, that is a regulatory molecule involved in things like coagulation, and the cardiac function is impacted by that. If we now instead have a vaccine, which is going to be a small defined amount of material put into your muscle in your arm, it's not going to replicate. So you're not going to have a fully fledged infection, just a small local reaction that will eventually give you an immune response. You don't also have those active mechanisms that the virus uses to avoid the immune system. So there's a hope that when you put in just the spike of the virus, you can generate an immune response without the virus being able to fight back and without a lot of those side effects. And that this should actually give you a better immune response even than natural immunity if we can get that vaccine right.
Okay, thanks, Tania. So for JP, after the approval of a vaccine, what will be the next steps?
Yeah, I think it's very important for people to realize that the approval of a vaccine, which we're all waiting to hear the news that we are seeing signs of efficacy and it is approved. It's important to realize all the steps that happen after. And of course, that is, the distribution, the manufacturing, the implementation in public health systems, the deployment. But importantly, when we will see the first set of approval, it will be for use in a given population at a given regimen. Very closely behind are other clinical trials that are looking at different populations as well, so in children and elderly and immunocompromised and so on, to determine and try to expand the parameters of use. And so that will continue to also happen. The route of administration, is it going to be intramuscular? Is it going to be intranasal? Is the dose going to be different in a population versus another? And so, that will be, it's important to realize that there's no single event that ends the development of a vaccine. It continues. And there's also this concept of phase 4, where after the vaccine is deployed in the population, of course, we will continue to monitor its efficacy, because now it will be deployed in much larger settings that was possible in a phase 3 trial. And we might be able to learn more about the immunity, the correlates of protection as well. And so that will continue to refine its use. And so a vaccine is very much, for lack of a better word, a living process, right? It evolves over time and gets refined, targeted, and how are certain populations with certain genetic predisposition, how do they respond to certain vaccines? And so all of that will be very important to continue. And so everybody is waiting for a single event of the approval of a vaccine. It's important to realize that it's an ongoing development process, and approval is one part of that whole process.
Thanks. So how do we ensure access of vaccines to all the people on this planet who will need one?
Yeah, I think that's a question that's on the mind of many people. I think in Canada, we are lucky to be able to count on a strong health system. And Canada's already announced advanced procurement of certain vaccine technologies. I think it's very important to realize that the strategy that is needed to curb a pandemic is very much worldwide. Borders are open. And so, everybody is going everywhere in the world, especially in this era. And so it's important to have a global strategy. And luckily, and I think this is important for everyone to realize, there are a lot of activities that are coordinated at the international level, not just for having national solutions, Canada or the US or Sweden, but there's actually organizations such as the WHO, Gavi, CEPI, the Gates Foundation, to name a few, who are trying to coordinate procurement such that if there is a commitment to buy a certain quantity of doses from the manufacturer, the bigger amounts of volumes allow for reduction in the price, such that countries that require some help to procure these doses are also able to procure in this way. And so there's a massive coordinated effort that is spearheaded internationally to make sure that developed and developing countries both have equal access to the vaccine resources that emerge.
Great, thanks. So for Jen, I have two questions. One is, how do I know the vaccine is working? And two is, how do I know the vaccine's safe? And I know there's a lot to unpack there, so I'll give you a few minutes to unpack those two questions there. Oh, yeah, I think you're muted still, Jen.
Good questions. Thanks for asking them. So I already mentioned this large phase 3 trial that gets rolled out in thousands of people. And so I mentioned one trial where there were 15,000 people in the vaccine group and 15,000 people in the placebo group. And so we'll know if this vaccine works because we'll see far fewer infections of SARS-CoV-2 in the subjects that got the vaccine as compared to the subjects who got the placebo. There are also some things we can measure. We call them correlates of immune protection. The most, I think, well described thus far is the generation of antibodies that neutralize the virus, neutralizing antibodies, which we know how to measure in the lab, but also T-cell responses that Tania mentioned. So some of these things will be also measured in people who got the vaccine, so that we have a sense that it's really producing the immune response we'd want. But we still lack a real, the neutralizing antibody is probably the best measurement we have to really say that the vaccine works, and of course that the people don't get the virus. In terms of how safe it is, so as I said, these are thousands of people. So we're gonna get a lot of information from these phase 3 trials on adverse events that would otherwise be rare and difficult to pick up. When an adverse event happens, it's obviously investigated really carefully. We need to understand whether that adverse event was a result of the vaccine or whether it was a coincidence and it happened to occur in someone who got the vaccine but for reasons independent of the vaccination. And so there are methods to investigate how that would be looked into. But as I mentioned, our biggest weapon here is to have just a lot of people so that we can get a lot of data and understand what are the rare side effects that we need to watch out for.
Great, thanks. So now we're going to turn to the questions from the audience. We've had quite a number of excellent questions. So I apologize in advance, I won't be able to ask all of them, but I'm going to try to get through as many as possible. So starting off, I'm going to ask this question to JP. What about the safety concerns regarding the presence of aluminum particles in vaccines? So that's question number one. And then question number two. So question number one is about aluminum particles in vaccines. And question number two is, COVID-19 is a disease with many facets, pulmonary, cardiac, renal, etc. Is there any evidence to suggest that an RNA-based vaccine would be associated with fewer or more side effects, such as stroke, than a killed virus vaccine?
Yeah, so there's a lot of content in these two questions. I can see that people are listening very closely. And so To answer the first question, I think the concept of adjuvants is one that's very important. I think we touched on it a little bit, during our opening remarks. And so when you have a very live virus, and that's what you're basing your vaccine technology on, a virus that replicates, maybe that's attenuated, so doesn't give the full range of symptoms, will allow the immune system to react very strongly to it. And so attenuated viruses are not one of the technologies that's the most advanced at the moment. And so basically, that would be the most efficacious type of technology. When you say, okay, I don't want a fully competent virus, I'm just going to take a small part of that virus and teach the immune system how to recognize that. For example, the S protein of the SARS-CoV-2, which is on its surface, which is what allows the infection of the virus to the host cells. And so if you're just giving a small part of the virus, such as this S glycoprotein, then the immune system has a harder time to mount a very strong response. And so the efficacy would be much lower, which of course is not what is desired. And so you need to add an immune stimulant together with your subunit vaccine, so the S glycoprotein, so that you can stimulate a stronger immune response. And so that's the concept of adjuvants. And aluminum salts is one of the oldest technologies that have been used in vaccines for a long time. There's a lot of data that exists regarding its safety and its immunomodulatory effects. There's other adjuvants that also exist that are being developed, already tested in the clinic, and they can bias the immune response one way or another way, more antibody mediated responses or cellular responses, depending on what type of immune response you want to elicit. Some of these are found from natural products that get included in together with the subunit vaccine. Some are synthetic components. And so the concept of adjuvant is very important, especially when we try to have components of the vaccine that are not the full virus, but just components of it to increase the safety. So that's for the first part. Tania, I see you're raising your hand. Do you want to comment as well?
Yeah, I just wanted to comment that to my knowledge, none of the phase 3 trials are actually using alum. The RNA vaccines, essentially their own adjuvant, because the RNA is seen as quite immune stimulatory. It looks like virus RNA, and it's encapsulated in what's called lipid nanoparticles, so it looks like a virus. It helps it get into cells. So, and then the other major contenders are based on an inactivated virus called an adenovirus that is gutted so it can't replicate, but it will express the SARS-CoV-2 S protein. And again, because it looks like a virus, the immune system recognizes it. And so there's no alum in that either. So alum is used historically in some much older vaccines. It is actually very safe, but it's actually probably not going to be used in the current batch of vaccines.
Great, thank you. Do either one of you comment on the use of the RNA vaccines, if they're going to have more or fewer side effects than traditional?
I can comment a little bit. So RNA is what they would call somewhat reactogenic because it's seen by the immune system as a danger signal. It's an infection, but they actually during the animal studies they actually modified the RNA somewhat to make it a little less immunogenic. And then in their initial trials, the dose escalation, they found a dose that had acceptable levels. And so what people will find if they take an RNA vaccine is they're going to be a little bit sore at the injection site because it'll create a local reaction. A little bit like if any of you took the 2009 adjuvanted pandemic flu vaccine, that was a much stronger local reaction, at least for me, than the regular seasonal vaccine. So my guess is we're going to see that a little bit with RNA vaccines. I think it'll make them very good vaccines, but you might have a sore arm and even a small fever with them based on what we've seen with the trials. But it doesn't mean it's dangerous or infectious. It's just giving a good local reaction.
Right. Yeah, it's proof that the vaccine is actually working in your body. That's right. Okay, here's a question for Jen about durability of immunity. So what, if anything, is known about the relationship between immune response, immunity, and infection in people who may have mild disease but long haul effects of months and months, as is emerging? So we're talking about people who have mild disease, but they have symptoms that are going on for a while, like, many months.
Right.
What is the relationship between the immune response, immunity, and infection in these people?
Right. So most of the studies, I think, are on people who are severe, moderate, mild, or asymptomatic. I don't know of any studies off the top of my head who specifically looked at what's being called long haulers. But what we do know about people with mild disease or even asymptomatic subjects, initially there was a paper that came out that said that their antibody responses diminished quite rapidly. There's been a flurry of papers since then. Most of these are still in what's called preprint, so they remain to be peer reviewed. But most of them agree with each other that at least over about a 4-month period, we make a sustained antibody response. In some papers, such as one from Florian Kramer's group in New York, has said that we also make a sustained neutralizing antibody response. And so we don't know why there was a difference between this early study and these later studies. I suspect it was partly technical, but the good news is that most of the people we measure do make a strong immune response in terms of their antibodies. But I have to say I haven't looked specifically into the long haulers that the questioner is asking about.
Okay, and here's another question, related to concerns about reinfection. Is herd immunity plausible given recent studies suggesting waning antibody response to infection? Is it more likely that we're looking at partial immunity, where we are likely to get re-exposed, but hopefully with less severe illness?
Yeah, maybe. Oh, is that for Jen or?
I don't know who it's for.
It's either of you is fine. Yeah.
I've already talked about weaning immunity, but.
Okay.
I think it's also important to understand the difference of different types of immunity. There is immunity that's called sterilizing. And so that basically puts a wall so that if you're exposed to the virus, you're not going to get it. That's one type of immunity. It's the most desirable, of course. If you can achieve sterilizing immunity, you're in good shape. A lot of times, as the other panelists were saying, this is achieved through neutralizing antibody responses. But there's a second type of immunity that's also very, very good, where you could get reinfected, but your body would be able to clear the virus very well, so it doesn't cause disease or it doesn't have a high mortality rate. And I think we need to also understand that that is a desirable type of immune response that we might want in natural infection, of course, but also through vaccination. And so, different outcomes to look for, but both very desirable.
And if I can just add, what vaccination does is it helps to even the playing field. Everyone gets the same dose in a controlled way. And so when you have a virus that's now infected 25 million people, all sorts of different doses, all sorts of different circumstances, all sorts of different people, you're going to see all sorts of different weirdness. But the goal of a vaccine is to really trigger the immune response in a predictable way so that we can generate what's called memory. So that if we are seeing the virus again, we can respond very rapidly and have very mild, if any, symptoms at all.
I guess I could add to what the two of you have said is that there's been some thought that, to completely prevent infection, we need those really good antibodies to be right in our places where we're exposed to the virus, in our nose and throat. And some vaccines which are given intramuscularly might give you circulating antibody that doesn't get to those surfaces high enough levels to actually completely prevent infection, but it would prevent you getting the pneumonia and the severe disease. So that would be good for the individual. What we don't know, I think that even some level of immunity will lower the time that you have active virus replication. So the hope is you'll also cut the spread. And there's actually been very good studies on influenza vaccines from our colleague Mark Loeb at McMaster University that did a study of the injected inactivated flu vaccine. And it not only protected 60% of those in that year who were immunized, but the older people in the community who didn't take the vaccine were protected 60% too. So I think there's very good evidence that vaccines will give us some level of herd immunity, even if it might not even be an ideal vaccine. It just needs to give some reduction in a significant proportion of the population.
Mm-hmm. Okay, here's a question for Tania about the natural versus vaccine immunity. There have been reports, mostly preprint, regarding potential systematic and long-term impacts of infection. Are these also a risk of gaining immunity naturally that are not present from vaccine-based immunity?
So you're asking me if the side effects, about side effects, or are you asking me about quality of the immune response? Sorry, I didn't quite catch that.
Yeah, I'm just trying, I'm reading, I was reading it verbatim and I'm trying to understand it myself, actually.
Find that.
I think the question was, are some of the long-term effects of COVID-19 going to be potentially experienced with the vaccine?
I would say that would not likely be the case, because don't forget, the vaccine isn't distributing all over your body. It's given very locally, and local cells of the immune system are going to respond to it and then send out lymphocytes to circulate and check for the virus. But you're not getting virus, highly replicating. I think some of these, what you call the long haulers, we don't actually know if they're, a lot of their symptoms may be from having that really, a lot of damage from the virus that lingers. But the vaccine is just not going to affect cells all over your body like the virus did. It's just not as extensive. It's a very local initial stimulus that then sends out the soldiers of the immune system to monitor for infection.
Yeah. So, and if the potency of an immunization is stronger than a given virus, could it lead to a stronger strain down the road? So, for example,
yeah, you're asking if there's some sort of selection. So, for example, what happens with influenza, we know that there's sort of this pressure. So there's antibodies developed against the tip of an influenza virus so that it can't infect. And that kind of drives mutations that escape. And in some ways, you may be selecting in a Darwinian way for these escape mutants. The thing about SARS-CoV-2 is it doesn't have nearly the mutation rate of influenza or HIV. So a lot of people think because it has a relatively, it copies itself fairly accurately, it doesn't make a lot of random mutations, there's a good chance that we're not going to see a lot of variability. There have been some genetic changes. From one continent to another, the virus has a few tiny sequence differences, but it doesn't change nearly as much as, say, influenza, which can change from the beginning of one flu season to the next and escape the vaccine.
Yeah, and I would add to that it's important to have a vaccine that's multi-pronged in its the way that it works. So if you're putting all your bet on antibodies that target a specific site, and there is a mutation that occurs, although Tania is 100% right that the mutation rate is much lower than from some of these other pathogens for which it's been very challenging to create a vaccine. If you're doing only on one site and just relying on the antibody response, then it's very risky. But most of the vaccines that are in development, of course, include much more surface of the virus and other regions that can be targeted. And the immune response that gets mounted is one that's very polyclonal. So some antibodies will, react to this site, others to this other site. Of course, there's a T cell as well that come and recognize different types of epitopes. And so as long as the vaccine is multi-pronged, then there's a good chance that it will be able to really keep the virus in check.
Okay. Yeah, I think, so these are the questions. So I have one question. We've been trying to develop an RSV vaccine for decades now. and respiratory syncytial virus. We don't have a vaccine against that. So how is it that we're able to develop a vaccine against SARS-CoV-2 so quickly?
Well, we don't have it yet. We have to wait for phase 3 trials, but it has been an unprecedented effort. It is a virus that's not particularly variable. RSV isn't either, to my knowledge. RSV is kind of a peculiar because, in the 1960s, there was a vaccine that actually made the disease worse because it induced the wrong kind of immune response, and people didn't understand that at the time. I think we have a much more knowledge now of how to test vaccines. There's a lot more approaches available now to us than just killed virus, which was the original RSV vaccine. So I think I'm optimistic, but we still need to actually see the results of phase 3 trials to know if it protects. I think the promising thing has been, there's been studies in macaques that say macaques that recover from SARS-CoV-2 are protected. And so there's very good evidence that protective immunity exists. And I think we have the tools and the modern approaches now to develop a good vaccine.
Okay, thanks. So here's a question about antibody-dependent enhancement. So we haven't seen a signal yet, but are the numbers too small until the phase 3 data? Can you comment on that?
Well, antibody-dependent enhancement is when an antibody binds in such a way not to completely protect against infection, like blocking the binding site, but actually enhances uptake of the virus. And the type of cells that take up the virus based on antibodies are things like macrophages. They're phagocytic cells that can grab the other end of the antibody and use it to pull the virus in. But from what we know of SARS-CoV-2, the cells that it replicates in are actually not macrophages, they're epithelial cells, and those are not phagocytic cells. So for this kind of virus, it's relatively unlikely that antibody-dependent enhancement would give you worse replication. Perhaps it could get into macrophages and cause some inflammation, but it's not going to do, for example, I think with dengue virus, it's a serious problem. But I think that really is in the case where the macrophages can be actively infected. You can correct me if I'm wrong on that.
Yeah, and I think that one aspect that's important to understand is that, of course, researchers are in focus, vaccine developers now, but, there's a lot of efforts that have been happening in previous years. And of course, SARS-CoV-2, which causes COVID-19, is not the first coronavirus that's around. There was a lot of research done during the SARS outbreak as well, SARS-CoV-1. And, some of the initial reports of antibody-dependent enhancement were reported in the previous context, although the literature was mixed. And so depending on which studies and how you set up the system, there might have been a signal, might not have been a signal. And so, what that taught us was to be mindful and really on the lookout. Is this going to be an issue? We should look into the data that emerges. And indeed, I find the scientific community has been extremely vigilant in looking for ADE, antibody-dependent enhancement. And the signs at the moment are that it's not a prominent effect, but we will continue to monitor it also in the context of vaccines. Everybody is very mindful of that.
Okay, here's one more question for the audience. When a vaccine is considered effective, past the phase 3 and approved, does that give us any information about whether you can still be contagious to someone else who has not been vaccinated? If not, is that tested at any point?
I'm happy to, circle back maybe to the question or to the comment I made earlier about, how does the vaccine work? Is it sterilizing immunity? So if you took the vaccine, you will no longer get infected, at which point, of course, if you don't have the infection, you can't transmit it. Or is there going to be a component that is sterilizing, but if you do get infected, then at least you have the immune system ready to fight the infection, to lower maybe the circulating virus, lower the titer so that you're less infectious. And so I think, depending on the mechanism of action of the vaccine, it will, impact the transmissibility of people who have been vaccinated. And so that'll be super important to continue monitoring down the road. And that's the ongoing life of the vaccine that I was describing earlier.
I think the primary endpoints of the phase 3 trials generally are the randomized, you either got placebo or vaccine, and did you get infected? But then I think as you start rolling out the vaccine, there needs to be secondary epidemiological studies, you know, our household contacts getting infected if they want it vaccinated, and that will need further study. But I think even if you only partially lower viral load, you may at least decrease the amount of transmission.
If I can just jump in, one of the hallmarks of an immune response is the development of what's called memory. And what memory does in your immune system is that if you are reconfronted with this virus, you'll respond more quickly and you'll respond more robustly. So if you're vaccinated, you should develop a memory response to that virus. And Jean-Philippe has rightly pointed out that that might end up being neutralizing, so done, virus is gone, or it might be that you'll slow down the replication of the virus. But one of the things that's been really tricky about SARS-CoV-2 is that there's this pre-symptomatic phase where you're very infectious. And that's a phase when your immune system is slowly just getting up to speed. If you're vaccinated, then in theory, your immune system comes up to speed much more quickly. And so hopefully, and we don't have any data to support this yet, but hopefully a vaccine will allow us to respond very briskly so that that asymptomatic period where the virus is replicating, hopefully you'll have already mounted an immune response so that you can really slow down the proliferation and dissemination of the virus.
Great, thank you for your, such great responses. So we're gonna go to our last question. This is, I think, the question that everyone wants to know the answer to. When do we think a vaccine is going to be approved? And I'll give each of you a chance to answer this. Let's see, we can write down our responses and see who's, we'll do like a pool as to who's got closest to what happens. Anyone want to jump in first?
I guess I mean, this is obviously dangerous to make these predictions, but if we just go by the numbers, we have 135 candidates in preclinical trials, 21 candidates in phase 1, 13 candidates in phase 2, and 8 candidates in phase 3. This is mind-blowing that we have this many vaccine candidates to deal with SARS-CoV-2 infection. So I guess if you're playing the odds, some of them are going to work, and the phase 3 are going on right now. So I'm hopeful that that in 2021 we'll have a vaccine. How long it takes to distribute it is really going to be key, and we have to be smart about how we distribute it so that we can protect the people who are most vulnerable.
I would also say that the phase 1 and 2 trials had very good antibody responses and T-cell responses for those first 3 front runners that are well into phase 3 now. So there's no question that we can induce an immune response. What we don't know yet is how much of an immune response is going to be associated with protection and how long it lasts. So I think I'm optimistic that the vaccine will give some level of protection for a period of time. What we don't know is if it's going to require annual boosting, or if you'll need maybe a couple of doses and then you have lifelong immunity, and that's going to take time. But at least it should buy us time that if we all get immunity for several months, it can start to break the cycle of infection and the pandemic.
Yeah, and I think, I'm also optimistic for the timelines that Tania and Jennifer are describing. I think, a vaccine that emerges that's 100% efficacious, is unlikely. And so I think, there's going to be levels of efficacy, levels of protection, levels of reduction of disease. I think when it becomes very critical is not just to look at the scientists and say, can you deliver a vaccine that's 100% efficacious? I think at one point the question shifts and say, okay, we have a vaccine that is showing good efficacy. Now, can we implement it in the global population? Is everybody willing to, do their role and take the vaccine to protect themselves at some high efficacy, but also to protect others? And I think that's the discussion that's important to start having at the moment. and that everybody continues to play a role in that pandemic, such that when those vaccines start emerging that might not be perfect in terms of 100% efficacy, but if they are, you know, at the 40%, 60%, 80% efficacy, if we all contribute and take the vaccine together, then that might be sufficient for to curb this pandemic.
I would like to add also that the manufacturing and who gets the vaccine is going to be a pretty important issue. So the RNA vaccines have the advantage that they're chemically produced. They don't need you to grow virus or cells, and they can make billions of doses, but it's a relatively unstable molecule. So it's going to require a cold chain, which for some parts of the world will be more difficult than others. And then we know that as you get older, the most vulnerable group to COVID is the over 70, over 80. They don't make as good an immune response. Are they going to need a higher dose of vaccine? So I think what public health authorities are going to have to figure out is, who do you give it to first? First of all, obviously, a lot of people think healthcare workers and those on the front lines, but then do you give it to the older population because they're vulnerable? But if they're not making a good response, do you give it to those around them instead? So there's going to be a lot of decision making once we have a vaccine in terms of logistic of getting it to people and who to give it to first as well.
Thanks so much, everyone. Unfortunately, we've run out of time. I just want to take this last second to thank our panelists who've done such a fantastic job. It's a great review of immunology since my last immunology course, like around 25 years ago. And thank all the participants on the webinar. And last but not least, I want to thank Elizabeth Loftus and Shaza Fadel from the center for organizing all of this and doing all the behind the scenes work. On behalf of the CVPD, I want to thank all of you, and we hope to see you for our next webinar. We've got a series of webinars lined up in the coming months, so we hope to see you again soon. Okay, take care, everyone. Stay safe.